Biochemistry 401 Lecture 21. Today we're going to talk about the electron transport chain. We're going to begin with a discussion of how reducing equivalents get from the cytoplasm to the mitochondrial matrix so that they can be used in the electron transport chain. We're then going to talk about oxidative phosphorylation, and we'll start with an overview of this process. We'll talk specifically about the electron transport chain and about integral membrane electron carriers and mobile electron carriers that make up this electron transport chain. We'll then follow electrons through the electron transport chain to see how this works. And so, how do reducing equivalents get from the cytoplasm into the mitochondrial matrix so that they can be used in the electron transport chain? Well, it's not actually the carrier that is transported, just the electrons, and so we're going to hand off electrons from one electron carrier to another. We'll start with an electron carrier that is in the cytoplasm that's reduced, and then we'll pass those electrons to a mitochondrial electron carrier and reduce that. So the electrons will be passed from the cytoplasm to the mitochondria by way of electron carriers. The electron carriers themselves don't cross the membrane, but the electrons do. There are two main ways that reducing equivalents get from the cytoplasm into the mitochondrial matrix. This is the glycerol 3-phosphate shuttle and the malate aspartate shuttle. The glycerol 3-phosphate shuttle uses cytoplasmic NADH and mitochondrial FAD. Now this mitochondrial FAD is shown here as EFAD because this FAD is actually a prosthetic group that's bound into an enzyme, and we'll look at this in a little while. With the malate aspartate shuttle, we begin with NADH carrying the electrons in the cytoplasm, and we're going to pass those over to an NAD+ molecule that's in the mitochondrion. We end up with NAD+ in the cytoplasm and NADH in the mitochondria. So now let's look at how these transporters work. This is a glycerol 3-phosphate shuttle, and it's known as the glycerol 3-phosphate shuttle because that is the molecule that's going to be oxidized to pass electrons to an electron carrier that has close links to the inner mitochondrion. This is an enzyme that is intimately associated with the inner mitochondrial membrane. It sits on the cytoplasmic face of the inner mitochondrial membrane, facing the intermembrane space. Now this process is going to happen in 3 steps. The first thing we're going to do is we're going to reduce dihydroxyacetone phosphate to yield glycerol 3-phosphate. And so the reaction is dihydroxyacetone phosphate plus NADH plus a proton yields NAD+ plus glycerol 3-phosphate. Dihydroxyacetone phosphate is being reduced, and NADH is being oxidized to NAD+. Now these electrons from glycerol 3-phosphate can be handed off to another electron carrier. This is bound into an enzyme, the mitochondrial glycerol 3-phosphate dehydrogenase. We're going to reduce FAD that's bound in this enzyme to yield FADH2, and in the process, glycerol 3-phosphate will be oxidized to form dihydroxyacetone phosphate again. But this shuttle doesn't stop here. The electrons that are carried by FADH2 can be passed off to a mobile inner mitochondrial membrane electron carrier that is known as ubiquinone. Now ubiquinone is sometimes called Q10. This is a mobile carrier that is lipophilic and shuttles back and forth in the inner mitochondrial membrane, carrying electrons from one electron carrier to another. Ubiquinone is often abbreviated Q. This is the oxidized form of the electron carrier, whereas QH2, ubiquinol, is the reduced form. We will look at this more closely in a little while. This is the malate aspartate shuttle. It is much more complex than the one we just looked at, and we're going to follow this really slowly so that you can figure out what's going on. The enzymes that are involved in this are a malate dehydrogenase in the cytoplasm, an aspartate aminotransferase in the cytoplasm, a malate dehydrogenase in the mitochondrion, and an aspartate aminotransferase in the mitochondrion. Now, both of these mitochondrial enzymes are in the mitochondrial matrix. And so we're going to start off with oxaloacetate in the cytoplasm. And we're going to reduce that to form malate. As we do this, the electrons from NADH will be transferred to oxaloacetate to yield malate and NAD+. So oxaloacetate is reduced to malate, and NADH is oxidized to NAD+. Malate will leave the cytoplasm and go into the mitochondrial matrix. Once there, it will be oxidized again to form oxaloacetate and reduce NAD+ to NADH. And so now we have our electrons transferred from the cytoplasm into the mitochondrial matrix. And that's what we want. However, what we don't want is we don't want a lot of oxaloacetate in the mitochondrial matrix because this might slow up the TCA cycle. And so we're going to move this oxaloacetate out of the mitochondrial matrix. But wait a minute, we can't do that because we can't transport oxaloacetate directly out of the mitochondrial matrix. There's no transporter. So we're going to use another one of our tricks, and we're going to add an amino group to oxaloacetate. Now remember, at the beginning of the semester I said it's important to know which amino acids have 4 carbons and which have 5. And I said it's important to know which metabolites have 4 carbons and which have 5. And this is why. Oxaloacetate has 4 carbons. So does aspartate. And so now what we're going to do is we're going to take an amino group from glutamate and transfer it to oxaloacetate. When this happens, oxaloacetate will be changed to aspartate. There are 4 carbons in oxaloacetate and 4 in aspartate. Aspartate can now leave the mitochondrial matrix by way of a transporter. But let's go back to the mitochondrial matrix for a minute. We took the amino group off of glutamate. This is the alpha amino group. And when we do this, we change glutamate into alpha ketoglutarate. 5 carbons in glutamate, 5 carbons in alpha ketoglutarate. This is the same intermediate that we saw in the TCA cycle. So we don't want this to build up either. And what we're going to do is we're going to transport this out of the mitochondrial matrix by way of a transporter. Once this alpha ketoglutarate is in the cytoplasm, it can accept an amino group from aspartate to be turned back into glutamate to begin the cycle again. And aspartate, once it gives off that alpha amino group, is changed from the 4-carbon aspartate into the 4-carbon keto acid, oxaloacetate. And in this way, this cycle is perpetuated. Take a minute to look at the arrows, and please note that when you see the transamination, what we're really doing is we're crisscrossing those arrows. So an amino group is going from glutamate to oxaloacetate, and oxaloacetate is becoming aspartate, and glutamate is becoming alpha ketoglutarate. And just to make it a little more clear, let's look at the amino transfer that's happening in the matrix. We'll start off with oxaloacetate. It is going to pick up an amino group from glutamate to yield aspartate. So oxaloacetate plus an amino group yields aspartate. Glutamate minus an amino group is going to yield alpha-ketoglutarate. And you can see the crossing of the arrows to represent where glutamate is going and where oxaloacetate is going. Aspartate aminotransferase works by way of an ordered bimolecular reaction. And what this means is these reactants engage with the enzyme in an ordered manner. First, aspartate is going to come in and it's going to lose an amino group. This amino group is going to become attached covalently to the enzyme itself. And in so doing, we're going to be left with oxaloacetate and an amino group that's covalently linked to the enzyme. Oxaloacetate leaves, and now in comes the second substrate, alpha-ketoglutarate. Alpha-ketoglutarate comes in, and it accepts the amino group from the enzyme. This causes alpha-ketoglutarate to become glutamate, and glutamate leaves. Both oxaloacetate and alpha-ketoglutarate are alpha-keto acids. But what does this mean? This is an alpha-keto acid that has 3 carbons. It's an alpha-keto acid because the ketone is in the alpha position relative to the carboxylic acid that is shown here. And so this is an alpha-keto acid. Likewise, a beta-keto acid has the ketone group at the beta position, and a gamma-keto acid has the ketone at the gamma position, and these are all relative to the carboxylic acid. You should know the identity of each of these alpha-keto acids. Can you identify them? Well, the 3-carbon alpha-keto acid is pyruvate. The 4-carbon alpha-keto acid is oxaloacetate. And finally, the 5-carbon alpha-keto acid is alpha-ketoglutarate. You should be able to identify these structures. This is going to be important now and also later in the semester. And so reducing equivalents like NADH and FADH2 are produced in such pathways as glycolysis, fatty acid catabolism, the TCA cycle. These are all oxidations of fuels. And now that we have these reducing equivalents, what are we going to do with them? Well, these 2 electron carriers have high electron transfer potential. These electrons can be transferred from carriers of high electron potential to those of lower and lower and lower electron transfer potential. And in so doing, as these electrons are passed from carrier to carrier to carrier, energy is released. And so, as these electrons are passed on through the electron transport chain, energy is produced that can do work. This work is going to be the pumping of protons from the mitochondrial matrix into the mitochondrial intermembrane space. This is going to set up a chemiosmotic gradient that will help to power ATP synthesis. And so what is electron transfer potential? Well, this is something that can be quantified through the use of the expression delta E prime sub zero. This refers to a molecule's reduction potential. And so we can relate reduction potential to the change in free energy under standard conditions at pH 7, because we are biochemists after all. And we come up with this particular equation, where N is the number of electrons and F is Faraday's constant, which is 96.48 kilojoules per mole per volt. Here's a table which is similar to that which we saw when we looked at phosphoryl transfer potentials. Please do not memorize this slide. You do not have to memorize a single thing on it. All you need to know is that the reductant, NADH, has a higher electron transfer potential than does water, and you also need to know the more negative the reduction potential. The greater the electron transfer potential is for the reductant, and the greater the tendency is to donate electrons. The more positive, the greater the tendency to accept electrons. And so let's look at this table, especially let's look at NAD plus and NADH, and oxygen and water. NAD plus and NADH are the oxidant-reductant pair, and this has a reduction potential of negative 0.32 volts. This means that NADH is a potent reductant and will reduce anything that is lower on this table. And oxygen is at the very bottom of this table. It has an electron transfer potential of positive 0.82 volts, and so it has more of a tendency to be reduced. Let's look at this in action. 1/2 O2 plus NADH plus a proton yields water and NAD+. We're going to split this into half reactions so that we can use the table above. 1/2 O2 plus 2 protons plus 2 electrons yields water, and the electron transfer potential for this is a positive 0.82 volts. NAD plus a proton plus 2 electrons yields NADH, and the electron transfer potential for this reaction is negative 0.32 volts. But the thing is, in the reaction above, in the full reaction, we don't have NAD plus on the left. We have it on the right as a product, not as a reactant. And so we're going to flip the half reaction, reaction number 3, to yield the half reaction number 4, NADH yielding NAD plus a proton plus 2 electrons. And so since we flipped it, we're going to flip the sign as well. And the electron transfer potential is positive 0.32 volts. And so now let's plug this into the equation using reaction 2 and reaction 4. The change in standard free energy is equal to minus the number of electrons times Faraday's constant times the change in reduction potential. For the first half reaction, we get minus approximately 158.23 kilojoules per mole. And for reaction number 2, we have the change in standard free energy is equal to minus 2 times Faraday's constant times 0.32 volts, which is equal to minus 61.80 kilojoules per mole. And so the sum is negative 220.03 kilojoules per mole. That's a lot of energy to be given off at one time. It is this reaction that we're going to do in the electron transport chain. We're going to transfer electrons from NADH and from FADH2, and we're going to give them to oxygen. If we were to do this all at once, we would yield a great deal of energy all at once. You run the risk of some of this energy being transferred to heat and to entropy. And so, instead, we're going to transfer these electrons from one electron carrier to another, and at each step, we're going to harvest the energy that's released by pumping protons from the matrix to the intermembrane space to set up a concentration gradient and an electrochemical gradient. It is this chemiosmotic gradient that is going to power the production of ATP from ADP. This table shows the electron carriers that we're going to use in the electron transport chain. You can see that NADH, the reductant, has the highest electron transfer potential. Then FADH2, cytochrome B2, ubiquinone, cytochrome C, iron, and water. And so we're going to pass electrons from a compound with the highest electron transfer potential to those with lower and lower and lower electron transfer potentials until we finally get to molecular oxygen, which will be the final electron acceptor, to yield water. In this slide, we see a schematic of the electron transport chain. NADH is at the top of the list. It has the highest electron transfer potential, and it's going to donate electrons to NADH Q oxidoreductase. This enzyme is also known as complex 1. The next is FADH. This can come through the glycerol 3-phosphate shuttle or from succinate dehydrogenase. Succinate dehydrogenase is also known as succinate Q reductase, complex 2. Now the electrons that reduce coenzyme Q come from 3 sources, as is shown here. They come from complex 1, they come from complex 2, and they come from the glycerol 3-phosphate shuttle. Coenzyme Q then shuttles those electrons to complex 3. And this complex acts to transfer the electrons from coenzyme Q to cytochrome C, so that coenzyme Q is now oxidized and cytochrome C is reduced. Complex 3 is also known as Q-cytochrome C oxidoreductase. Now that we have the electrons in cytochrome C, we're going to use those electrons to reduce molecular oxygen to form water. And the enzyme that does this is actually complex 4. Complex 4 is also known as cytochrome C oxidase because we're oxidizing cytochrome C. And so this schematic shows the electron transport chain. The large complexes, shown in yellow, are integral membrane proteins, and these are complex 1, 2, 3, and 4. 2 are mobile carriers, coenzyme Q and cytochrome C. Coenzyme Q is also known as the Q pool because this consists of both ubiquinone, which is the oxidized form, and ubiquinol, which is the reduced form. This Q pool resides in the inner mitochondrial membrane and shuttles back and forth in a lipophilic environment, whereas cytochrome C operates in the intermembrane space. So now let's follow the electrons through the electron transport chain. We're going to start off with NADH and with FADH2. NADH will reduce components within complex 1, and in so doing, the energy that's released will be used to pump protons from the mitochondrial matrix into the intermembrane space. We can also use electrons from FADH2 to reduce components in complex 2. Complex 2, however, cannot pump protons from the mitochondrial matrix to the intermembrane space. The electrons from complex 1 and complex 2, and also from the glycerol 3-phosphate shuttle, can be used to reduce ubiquinone to ubiquinol. In this diagram, we see coenzyme Q, which is also known as coenzyme Q10. This hydrocarbon tail makes it highly hydrophobic. On the left, we see ubiquinone, which is a fully oxidized form of coenzyme Q10. This is a quinone with 2 double-bonded oxygens as substituents of the central ring. We can add 1 electron at a time to form semiquinone intermediates, shown in the diagram in the middle. When coenzyme Q is fully reduced, it is known as ubiquinol, and it is a diol having 2 hydroxyls that are bonded to the central ring. Ubiquinone, and of course ubiquinol, have a long hydrocarbon tail that is shown here. So, which one is this? Ubiquinone or ubiquinol? Well, this is ubiquinol, and we can tell because it's a diol. There are 2 OH groups that are shown here, and this is the reduced form of CoQ10. And if you got this right, very good. Now this CoQ pool carries electrons from complex 1 and 2, and also from the glycerol 3-phosphate shuttle, to complex 3. And this complex will pump protons from the matrix to the intermembrane space. Next, these electrons can be passed to cytochrome C. Cytochrome C will pass on the electrons to complex 4 to reduce components within complex 4. As these electrons are passed, The energy that's released will pump protons from the matrix to the intermembrane space. And oxygen will be the ultimate electron acceptor. Complex 4 must hold on to this oxygen and reduce it and release it as water. There are great measures to make sure that this oxygen is kept in check until it's time to be released. Just like with hemoglobin, we want to make sure that we do not release any superoxides. Reactive oxygen species such as this are bad news for biological systems. For this reason, oxygen is held in place as a peroxide, bridged between an iron and a copper metal ion. Through the addition of 2 protons and 2 electrons, 2 hydroxyl groups are formed, and then an additional 2 protons and 2 electrons releases 2 molecules of water. You do need to know how oxygen is held in place within complex 4 by the iron-copper center as a peroxide bridge. These next 2 slides are a series of notes for you to help you to study. You will need to know the information that's found on these 2 slides. In this slide, we see the working components of the electron transport chain. These are the types of centers and cytochromes that actually bind to the electrons. What you'll notice is that we see metals such as iron and copper, and we also see sulfur. Additionally, what is not seen here is the vitamin derivative FMN. This also helps to carry electrons within the electron carriers of the electron transport chain. Please do not memorize these structures. It is simply enough that you understand what sort of molecules are helping to transfer these electrons within the complexes and mobile carriers. And this marks the end of Lecture 21. Have a great day.